So this chapter, okay, first of all, before we get started, I always forget. Does anybody have any questions about anything we've learned yet? Any questions about anything in class? No. Okay. All right, chapter 13 is dealing with disease, basically, what it is, how it happens, how the microbes make it happen, and all of that. There's a few factors that could cause disease. Some of them are infection, right? But that's only one. You could have genetics, aging, and just malfunctions of your organs and all sorts of stuff related to that. So infectious disease is any disruption of tissue function caused by microbes or their products, okay? So any deviation from health counts as disease. And then it's caused by microbes. That's infection. So our normal biota actually plays a pretty big part as far as keeping us safe from the bad guys and all that. A lot more than you might think, actually. So your normal biota can be anything: bacteria, fungi, protozoa, and viruses. I do want you to notice that there is another thing that we have talked about and learned about in microbiology that is not up here, and that is the helminths, okay? The helminths are the worms, right? The parasitic worms. They are not part of your resident biota. I don't care how many TikToks you've seen of people, drinking cayenne and lemon, and then they got rid of all of the parasites in their body because they saw all the worms in their stool or whatever. That's all BS, guaranteed. What probably actually happened to those people is if they did take things like ivermectin, because a lot of them take like HeartGuard and stuff. I don't know if you guys have seen this, if you know what I'm talking about. This is a real deal. And they'll take like HeartGuard or whatever with ivermectin. It's incredibly dangerous medication. And then they're like, oh, look at the worms in my stool. Well, yeah, that's not worms, honey. That's the lining of your intestine. And that is a real thing. That can happen if your intestines get irritated. Even if you have ulcerative colitis or IBS, that can happen to you. As just part of those diseases. But yeah, that's all it is. They think it's worms. It's not. It's their own lining of their own intestine, which I'm just like, y'all, really? Like how many people a day do you think is getting a colonoscopy? And you think there's worms just hiding that nobody knows about? I don't think so. But anyways, not worms, okay? Bacteria, fungi, protozoa, viruses, the tiny guys. All right, some of them are beneficial. They actually can help us out. Some of them are just going to be, breaking down food and getting nutrients for themselves, and just, whatever, they're just there. So our resident microbiota plus us, that makes the holobiont. You guys remember the holo enzyme we just talked about in the last chapter was the whole enzyme, the apoenzyme plus its cofactor. Well, it's sort of like that here. The holobiont, the whole biont or biological organism, is going to be us plus our entire resident biota. That's what that term is referring to. Our normal biota can influence development of your organs. That's real. That usually has to do with how, if you have normal biome going on in your gut as a child and you're growing up, then you're more likely to have a healthy GI tract in general, let alone the good effects it can have on your immune system and training your immune system as far as what's a good guy and what's a bad guy and everything after that. I don't know if you guys know this, but one of the biggest things in science right now is looking into the effects of the gut biome on other aspects of your health other than your digestion. So things like mental health, very, very closely linked together, which is a shocker for a lot of people. But yeah, apparently, if you have certain kinds of microbes in your gut, they will break down certain kinds of foods, give certain kinds of products, and is more likely to give you a healthy mental, mood and everything like that, mental health. Whereas, if you have the other kinds that aren't giving you the healthy stuff, they're giving you maybe toxins or something like that, you're more likely to have a mental disorder, like even bipolar disorder or schizophrenia. I'm not kidding, it's that serious. So it's linked to that sort of stuff. That is pretty crazy to think about. So we're talking about an aspect of your body that's non-human that has a massive effect on you as a human. So there's that. They're there also, they help with preventing overgrowth of harmful organisms. My favorite analogy, of course, the stadium seats. So we have all of our good guys, our biota, taking up the stadium seats so that the bad guys can't get a foothold and start growing and causing disease. Okay? Microbial antagonism, that's an example of microbial antagonism. Microbes basically serving us, protecting us from the bad guys. They could be doing that just by existing there, or they could be doing that by making toxic products that are toxic against the bad guys and all sorts of stuff. All right, when do we start getting our biome? And when does that start getting associated with our body? Well, it actually starts in utero. They used to think that it was sterile in there, and they found out that you do have a biome in your, what is happening? What is, I have this, it was working. Whatever. You do have a biome, actual uterus. It's just, there's nothing good about this computer, I'm starting to think. I don't know where this is working. It's plugged in. I have no idea. Anyways, you do have a biome in your uterus. And I don't know, I'm not going to talk about what type of microbes. I don't need you to know that or worry about that right now. But there's microbes in there they didn't think there was. And turns out that's where your fetus is going to start getting exposed to bacteria. No surprise when you say it that way. Previously, it was thought that you started out with it whenever you were born. But now we know that's kind of the second stage of things. Going through the vaginal canal as opposed to being born C-section, you're obviously going to get exposed to different kinds of bacteria in those locations, right? Especially because usually if you're being born C-section in a hospital, they've tried to sterilize things as much as possible. So you're not really going to get exposed to many bacteria until, you know, start getting handled by healthcare workers later on, like after you're in, you know, your little pediatric ward and everything like that. And of course, the vaginal canal has all sorts of bacteria associated with it. You're going to get colonized by different bacteria based on how you were born. Then we have breast milk. Breast milk itself that you'll be drinking as a baby has a microbiome. That's no surprise. I know you guys know about this. They talk about this now all the time. So breast milk, not just colostrum, okay? But colostrum is going to have the highest concentration of antibodies provided from the mother, but that continues to go on for breast milk during the entire breastfeeding time period. Good, healthy microbiome, those organisms that are associated with breast milk break down sugars that you yourself would not be able to break down. So you're getting colonized by bacteria that can help you break down sugars. Or if you weren't exposed to that, you might be intolerant to those sugars otherwise. So that's the idea with the breast milk. I even had a student on my Monday, Wednesday class in the mornings where she was telling me she was homeschooled, but she had a cousin who, like, everybody in her family had pink eye. And so the mom took the breast milk and she put drops of breast milk in everybody's eyes and it cleared up. That's how effective it can be. I mean, it's like good stuff. It's good for you. Not going to cure everything, right? But that's the whole point. Now, if you're feeding off of formula, are you screwed? Not necessarily. You can buy formula that has probiotics in it. So you can still have your baby get exposed to a nice, healthy microbiome that way, okay? A lot of formulas are sterile. I will say that. You have to look for the ones that do have the prebiotics or probiotics as well. All right, caregivers. This is literally anybody handling a baby after that point. That makes sense. You know, kisses from dad or hugs from your uncle or whatever it is, you're being exposed. So we're not just talking here about necessarily your gut biome. Of course, now we're talking about your skin biome and everything else. And then the environment that you're going to be in. Yeah. What is this kid doing? Playing with the dogs? Yes. Puppies. That's what this baby's doing. And you can imagine the bacteria that can be associated with that. I love dogs. I have 3 of them. And I still, you know, when you put it in this perspective, it feels gross. So anyways, now we'll move on. We know that's colonization. That bacteria getting our biome, that's colonization. Now we're going to move on to infection and disease. Infection is where we have pathogenic organisms penetrating host defenses, entering tissues, multiplying, right? Eventually leading to disease. The problem with this, it's not a problem, but the whole thing I want to point out about it, it says pathogenic microorganisms. I mean, they even have the propensity to be pathogenic. They might not always be a pathogen. They might be opportunistic, okay? And then a disease is where you have any deviation from health is what the way I like to define it, and I think that's pretty accurate. But you're having a pathological state. You're having some sort of presentation of things going less than ideal, right? You're having damage, disrupting tissues. You have symptoms and signs, okay? That is disease. All right, here's throat. Okay, Yeah, this stuff is kind of obvious too, as far as things that can make you more susceptible to infection: age, genetic defects, immune issues, pregnancy, surgery, organ transplants, underlying diseases like cancer, chemotherapy and immunosuppressive drugs, physical and mental stress, and other infections. I'm sure none of this is really a surprise. If any of this is a surprise, feel free to ask me about it and I'll tell you why it would make you more susceptible to infection. But really, I feel like the bottom one, up from the bottom, the physical and mental stress kind of sums up why the others are a problem too, right? That's what we're talking about, physical stress, but in different forms, a lot of these. Endogenous infections. We're going to get into endogenous infections and exogenous infections. Okay. Endogenous. These are infections caused by organisms that are already in your body. The example that it gives, and like it says, it can occur when normal biota is introduced to a site that was previously sterile. The example that it gives is Streptococcus pneumoniae, often carried as normal biota, can cause pneumococcal pneumonia in AIDS patients. I don't like this example because it like downplays a Streptococcus pneumoniae, okay? Streptococcus pneumoniae, it'll like to hang out in like your upper respiratory tract. It's pretty normal there. Yes, obviously in its name, it even says it can cause pneumonia. It's actually the #1 cause of community-acquired pneumonia, okay? So no, I mean, it's not just going to be associated with pneumococcal pneumonia in AIDS patients, but it's very serious to those patients, right? Whereas you might get pneumonia from it and just have to take antibiotic and you're fine, So that's, I guess, why they're saying it that way. But anyways, my whole other point with Streptococcus pneumoniae is something that I'm going to come back to in a little bit. Streptococcus pneumonia isn't just the #1 cause of community-acquired pneumonia, which is just normal pneumonia. Your other option is healthcare-acquired pneumonia, okay? But it also is the #1 cause of meningitis, bacterial meningitis. Really, really by far the #1 cause, okay? And I'm sure you guys know and have heard that bacterial meningitis is no joke. It is actually a very serious infection. So serious that like if you were staying in dorms or something like that, they would absolutely require that you have been vaccinated for the pneumococcus because they don't want people getting pneumonia and it spreading amongst, people who are living in close quarters. Where it's the most likely to get spread. We do know that meningitis in general is kind of difficult to spread. It's not wanting to jumping all over the place all the time, like COVID, but still, it's a bad deal. When you do get it, it's a bad deal, right? It's a really dangerous, deadly disease. We'll come back to meningitis later. First, I want to get into whatever this is. Nope, it still isn't working. Great. I know that it has to be connected to it. I just don't understand this computer, I guess. Anyways. All right, so here's the normal person, whole biome, includes you and everything on you. So here's what can happen. You can have potential pathogens and transients, and they just go away. You get exposed to new guys or a part of your body. Maybe you touch this. I'm sure that this isn't sterile up here. The odds that I'm going to get sick from touching bacteria that were transferred onto here are slim to none, right? My system's going to kick them right out. So that's what it's talking about there. You get exposed, but it's just gone, whatever. Next option, the pathogen can become, or part of the pathogen and occasionally the resident microbiota, so things that were already in you or things from outside, either way, can lead to infection. So these organisms now have taken hold and multiplied, usually in areas they don't belong, or if they're normal pathogens, they're just being a pathogen. And anyways, most of the time, even these are going to be asymptomatic and clear. So it might start growing, but your immune system is going to fight it off before you even have any symptoms. Okay, so that's what that's talking about. But there's options. Otherwise, we have infection that will lead to disease. So you got the infection and it goes far enough that you start having symptoms and signs, and now you're going to have actual disease develop. Bad, you've got fever, maybe you've got the chills and all sorts of stuff associated with it. You'll experience the disease, and usually we will eliminate them either on our own or maybe even through antibiotics, but it's one or the other. And then if you don't do that and you just let it go too long, you can die. Or certain diseases, you don't have much of a hope, right? Like Ebola or something. Some of these, after you have kind of knocked down the disease aspect can become part of a holobiont, so part of your normal biota. This is stuff like Staphylococcus aureus or C. diff, because those can be just part of your normal biome, and you have to get it from somewhere. So that happens that way. Here are some examples. My nose could not be any more tickly if it tried. Okay. Here are some examples of organisms that are going to be found in certain areas of the body. And it even has them grouped up by gram-positive, gram-negative bacteria, fungi, protozoa, and whatever. So I don't need you to know any of this right now, but if you're interested, it's there. Leading into the infection. All right. Pathogens are microbes with a relationship with the host that is parasitic, causing infection and disease. Okay. Pathogens, microbes that cause disease and infection. True pathogens. These true pathogens are going to cause disease even in healthy people. Okay, even with a healthy immune system. This is like COVID, right? Even healthy people get this and it can cause disease in you. Next, we have opportunistic pathogens. These are only going to be causing disease and illness in somebody with their defenses being compromised or when they're introduced to a part of their body that they don't belong. Let me walk you through an example. I know it gave us that example of Streptococcus pneumoniae. That's nice for it. But like, I feel like a very more relatable one, especially for the ladies, is going to be a urinary tract infection. Okay. 80% of urinary tract infections, it might even be 90. I don't know. I know it's at least 80, are caused by E. coli. Okay. I don't know if you guys have been paying attention or not, but E. coli is in the colon. It's not supposed to be in your urinary tract infection, right? So here we're talking about a microbe that has moved from a GI tract into the urinary tract infection. If you think for a second that it was somebody else's E. coli, no, y'all, it's from your own booty. Okay, that's where it comes from. That's why they teach you to wipe from front to back, right? Because it's the back going to the front that's the problem. That's where you get your UTIs. And of course, if you're having sexual activity, that stuff gets mixed up. We all know. So you get your urinary tract infection. That E. coli does not belong in your urinary tract. So this is exactly what we're talking about with an opportunistic disease. It was fine in your GI tract. All right, let's now that we're getting I can't speak sometimes. Okay, now that we're getting introduced to the types of pathogens and what types of infections they may cause, especially if we're talking about the true pathogens, we can get a better sense of a biosafety level and what that means. Okay. You've probably heard about biosafety level, whatever, or we haven't really mentioned it much in our lab. But I'm going to tell you right now, it's pretty easy. It's pretty straightforward. And so usually they got, like it says, based on the degree of pathogenicity of the organism and the danger of handling it. How likely would you be to get that disease by working with it, right? So those things. This room right here, right now, would be biosafety level 0. Even the bathroom, I hate to say it, is a biosafety level 0, okay? Because we're not intentionally working with infectious organisms or even potentially infectious organisms at all. Okay, so obviously our lab in the micro lab upstairs, that is a biosafety level 1. We are not working with true pathogenic microorganisms. Yes, we're working with staph. Yes, we're working with E. coli. Yes, we're working with salmonella, but they don't have any of the genes that allow them to be pathogenic. Okay, you have to, that's this whole separate strain that you'd be dealing with. So they don't have any of those genes, or we're going to be dealing with bacteria that like grow in the soil or are normal, healthy, like Staphylococcus epidermidis or lactobacillus or whatever. They're just not that terrifying. So that's going to be open bench, no special facilities. Yeah, it says it's typical of most microbiology teaching labs, and access may be restricted. Like, we don't really have any true restriction here, but it's just generally known that like, people who are going into that lab in a micro lab are in microbiology and not just rando people. And that door is never unlocked when I'm not in there or there isn't a class going. That's just because more or less we don't want people stealing the microscopes. I don't know if you've ever seen Dr. Shearer talk about people trying to steal a microscope. If I leave his door unlocked, guys, for 10 minutes, he comes and finds me and he's like, you need to come lock this door. I'm like, okay, dude, I'm coming. I'm sorry. All right. But yeah, it is that serious. So anyways, next is level 2. We have The E. coli, salmonella, staph, like I was just talking about, but these are the pathogenic strains. Pretty easy to work with, pretty safe as long as you're wearing the proper PPE. We're talking about gloves, we're talking about lab coats, we're talking about special procedures as far as like cleaning your surfaces. I know we do use disinfectant, but there will be more involved there, right? Regular cleaning with bleach and stuff like that. Also, in our lab, we have that one red trash, the biotrash, right? We have that one biotrash, and everybody has to walk all the way over to it to put their trash in it. In biosafety level 2, that would be considered a hazard, having it that far away from people who would be working that much. So they would have to have one at every bench, right? So those are other things that to be considered, as well as signage and everything saying, hey, there's a restricted here, just biohazard signs and all that. Okay, next we have, so that's typical of what you might think of people working in a research lab. Yeah, what we normally think of. They're not wearing any like special hoods or anything. They're just normal people, lab coat, gloves, but they have a little bit extra precautions than we do in our lab. All right, level 3, now we have all of that, but we usually have a very restricted area. Usually there's an anteroom. An anteroom is just like a changing area or something like that, where you're going to be putting on PPE that's usually disposable, and you'll usually incinerate them before you go home or whatever. Sometimes there's even a shower that you'll have to shower before you go home as well, like go take a proper shower. Then also those labs tend to be negative pressure, so that if there is some break in like HEPA filtration or something like that, the air will rush into the lab as opposed to out. So we're protecting any environmental things. That's important because a lot of the labs that are working with tuberculosis are going to be BSL-3 labs. And so I even went and interviewed with a guy who worked on tuberculosis, and I got to see his BSL-3 lab at Berkeley. He did not like me because I didn't speak Spanish. I told him I was more than willing to learn Spanish, but he was just not having it. So so I didn't go to Berkeley. I went to Penn instead. But yeah, so I just remember that though. I remember them like having that changing area, all the disposable paper suits and the booties and everything like that to go work on it. And I could not go into the actual lab. It wasn't allowed because of I wasn't allowed to. Next, we have BSL-4. Now, if we think that that's crazy with BSL-3, where you're going to have to change your clothes and have the burning of the little paper clothes and all of this stuff, the positive pressure on the outside and negative on the inside, BSL-4 takes it to that high level of what you see with like Ebola and all that, right? So outbreak is what I'm trying to think of, like outbreak. You wear the suit that's like that heavy sort of rubbery suit, plasticky suit. You have rubber gloves that are pretty thick. You're going to be taping your gloves to your suit every single time you go into that lab, and your boots every time you go into a lab. They're not like some sort of special situation that seals themselves. No, you just use tape. That's what you do. You just make sure that it's clean. What you do is you will put on your suit and your boots and your little hood, and you connect everything and make sure it's all sealed up. And then you check it before you even go into the lab. Make sure there's no leaks. You always do it with a partner so that the partner can check you too. You check yourself, you check your partner. And that's how you always do it, going in and out of these places. I've never worked in a BSL-4 lab. There are actually very few. It seems like there should be a ton of them. There really are not. In the United States, not very many. So when I was working on my virus, which was a BSL-4 virus, according to the Army, it was BSL-3+, but to everybody else in the world, it's a BSL-4 virus. And, having to go see people who were working on that virus so that I could have some work for me on the actual virus, and I just used the parts. I got to talking to a lot of these people. One of the people that I worked the closest with was a CDC. So I got to go to Atlanta, and whenever they were building their brand new BSL-4 labs, they didn't even start using them yet. Nope, they weren't even working them at all. They're just cycling them on their cleaning cycles to make sure that worked. But we got to go into that lab because it was still clean, right? But yeah, they went through their BSL-3 and then into the BSL-4. So you had to already have gone through the procedures of the BSL-3 before you went there. They had retinal scans to get in there, like no joke. And it was really cool. And they went through, basically put on the suits, check it. Then you go into this little room. And it's a room, all the doors are closed, maybe fit one person in there. If you fit two, man, it would be tough. But you stand in there and wait for it to pressurize or depressurize, I guess, because it's negative pressure, just like the other, right? Very highly contained, so you're just waiting for pressure to change. You go in there, you do all your work with those big gloves, working the little micropipetters, all these little tubes, everything with these freaking big gloves and plastic in your face. You can hear yourself breathing every breath that you take. And you have to have special like speaker in your helmet so you can hear people talking to you at all. You're hooked up to air and all of this stuff, right? No, thank you. Then I was talking to them. I was talking to one guy, Eric, that worked there, and he was saying like it's not good. Like he's trying to get out of that line of work, and he was like, it's just you have to get used to it. And I was like, I bet because I would get super claustrophobic. You could put me in that suit and tell me to play with puppies, and I would get claustrophobic from that. It's just stressful, right? He didn't like it, and he hated that part of it. But he was the one that was doing all my work for me at the CDC. And then on their way out, they would go through a chemical rinse, and they would stand in a hot chemical rinse the entire time that they were, recompressing in that same chamber on your way back. Just stand in this hot chemical. Sounds such like a nightmare. So here, of course, we're talking about Ebola and similar things that can make you just bleed out and basically explode and transfer to other people. So it's no good, that stuff. Virulence is a term that's going to be referring to the degree of pathogenicity. Basically, we're going to, in the degree of pathogenicity, mostly we're going to be talking about the disease aspect of it, as far as the symptoms and the signs and the damage associated with it, and its ability to establish itself in a host as well. So anything that allows for that, so degree of pathogenicity, the amount of how bad that is, right? So that's what virulence is about. So virulence factor is anything that allows that to happen, basically. Anything that it uses to make that happen. Flagella, toxin production, antibiotic resistance, anything like that. Infectious dose is a term that's referring to the number of microbes you need to start an infection. So bacteria typically need more than one bacteria there because some of them are going to get killed by the immune system and da-da-da-da. Viruses often have an ID of like around 1. So you only need 1 viral particle to start a disease. A low ID for bacteria is one of the more infectious, but still it has to be active in order to be transmitted, is tuberculosis. You need 10 bacteria in order to transmit tuberculosis. But again, it has to be somebody active. But yeah, so that just puts it into perspective. Smaller infectious dose, like 1 virus, that's a greater virulence. Well, polymicrobial infections are not any surprise. We're talking about if you get the flu, how you're likely to get bacterial influenza as a result, right? Your immune system's down, your body's being stressed. That's the polymicrobial infection when you have that happen. So these are normal types of things, nothing crazy. All right, let's go through the steps involved in getting a disease. First, we'll start with the portal of entry, how the thing gets into you. There's all sorts of ways, going through skin, mucous membranes, inhalation, eating, all sorts of that sort of stuff. Sexual contact. It can come from outside or from inside. We've already mentioned the inside. Remember, that's endogenous. Endogenous, already there, maybe somewhere it shouldn't be. Exogenous, it came from outside of the body, like COVID or the flu. These things that aren't normally part of your body, and now you're sick. How did we get in the skin? I'm not going to go through each of these, y'all. How do you get in the skin? You either puncture it or you are something that can go through intact skin, like those worms we mentioned, right? Gastrointestinal tract, you usually have to go through eating or drinking. You have to be adapted to deal with the enzymes and the pH of the stomach. Respiratory tract, you have to breathe in through the nose or the mouth here, so cool. And then we have urogenital portals through genital area, usually sexually transmitted diseases is what we're talking about here. Okay, there are pathogens that will infect, be associated with pregnancy and birth, okay? This will usually have to deal with the mother being infected and transmitting it to the child somehow, either through the placenta or potentially just, direct exposure to the disease itself. So let's get into this. Transplacental infection of the fetus, we're actually talking about if the mother is infected, it can cross the bacteria or virus or whatever, cross that placenta into the baby's bloodstream. Now, just can, it has the ability, that's a virulence factor to be able to do that. Not all of them can. So here's a word, torch, that's supposed to be telling you about common infections of fetuses and neonates. I hate this because of the O, we'll see in a moment. So T is toxoplasmosis. This is a protozoan infection. This is the one that you get from handling kitty litter, that you're told not to do it. Severe defects in children, potential stillbirth and stuff like that. all sort of congenital abnormalities. Pretty serious, like it is that bad. Okay, the O means other diseases. So why are we splitting this up? I don't know. It makes no sense to me. Syphilis, I'll go through the ones they have listed. Syphilis, Coxsackie virus, that's like hand, foot, and mouth disease. You know, when the kids get the little rashes on their feet and their hands and stuff. Varicella zoster, that's chickenpox. HIV, it says AIDS, I don't like that. HIV. Chlamydia, okay? All right, moving on to R, this is rubella. You've probably heard rubella spoken by somebody, but never really thought about it after that. Okay, if you've been pregnant, one of the things that they will definitely ask you in the United States if you're pregnant is if you've had your MMR vaccine. And they'll be concerned about that because MMR, measles, mumps, rubella, will protect you from rubella infection. Okay, rubella is a virus that's probably going to give you barely any symptoms, if any, as a grown adult. Probably none, and almost all adults will have get exposed to it somehow. But you know, we have vaccines, so we wouldn't have even known. Anyways, rubella, it can lead to spontaneous abortion and stillbirths. That's a pretty serious one. Okay, cytomegalovirus, it can cause abnormalities in the fetus. CMV, they test you for it if you donate blood. You're either CMV positive or CMV negative. Most people are CMV positive. You've had it at some point in your life. It's just like a cold, but it's very bad. Like some of these other ones, they're not nothing for an adult, but pretty bad for a fetus. Yeah. And then herpes simplex virus. Do not underestimate the power of herpes during birth. Okay, it is no joke. If you're somebody who has herpes and you are not having an outbreak, right? You're just living your life. Maybe you take Valtrex, I don't know, but you're not having an outbreak and you go into labor, you're fine. If you are having an outbreak of herpes in the genital area while you are in labor and you're giving birth, your doctor will not let you give birth vaginally, no matter what your birth plan is. Okay? C-section right away if they see any evidence of herpes. Okay? Why? Because herpes exposure is bad enough whenever it gets into other aspects, just causing problems with skin and everything. The baby can't defend itself like adults can, right? But if it gets into the eyes, it can move very quickly into the nervous system. It's not uncommon. It really isn't. If you do get exposed to herpes as a baby being born. And that will lead to meningitis caused by herpes, which is a possibility. Like I said, not uncommon if you are in this situation. 80% fatality rate. Imagine giving birth to your child and then it dying because of your herpes infection, because maybe you didn't tell somebody, somebody didn't notice, or whatever it is, right? As far as herpes goes within adults, like that's your own business. You're taking Valtrex, you should probably notify people if you have it, but I don't care, just whatever. But during birth, that's serious and people should be aware of that, especially if you're going to healthcare. Yeah. All right, adhesion. So we got into our, victim. So now we got to stick down wherever we want to go. Find our location of where we're going to multiply. That's adhesion. For bacteria, we're often talking about fimbriae, right? Those little appendages that are not for movement, but look like they would be. Those appendages, fimbriae, that'll stick to things, as well as the capsule. We know that's sticky, that'll stick to things. And the slime layer, similar concept, okay? Sticky, we stick to things. And then we know that also can be used by bacteria to form biofilms, not least to quorum sensing. We know about biofilms and quorum sensing, their ability to talk to each other about the health of the biofilm, but this can also be used for establishing infection if there is biofilm involved. All right, so here are examples, picture examples of what I'm talking about. Fimbriae at the top, okay? Capsules next. So these are bacteria. So capsules are sticky and it sticks on, and then spikes of the virus. Remember those glycoprotein spikes of the virus meeting in like a key into the lock, the protein on the host cell. So that's some ways that they can get into where they're trying to go. Here's some other examples if you're going to try to learn about that. I'm not going over it. All right, so you got to the place you want to be, and now you got to avoid the body attacking you. Phagocytes, these are those white blood cells that eat things up. I keep saying about how capsules help evade phagocytosis, right? That would be considered an antiphagocytic factor, right? It's going to be the one that falls right here, makes it physically difficult for the phagocyte to engulf them. capsules. We also have some organisms that can kill the phagocyte itself. So they don't get eaten. And then we have some that are adapted to survive inside phagocytes after ingestion. The preferred location for multiplication of the tuberculosis bacterium is within alveolar macrophages. Those are macrophages are, by the way, those are phagocytes, specific kinds, that will hang out in the lungs, deep in the lungs. So they send, so your body says, oh, there's a bad guy in here. We need to get, call in for other forces to help us out. And then more forces get there as alveolar macrophages, because that's what you send in that area. And the bacteria is like, thanks, guys. Thanks for bringing more housing. That's pretty much how they feel about it. They like to grow in there. So that sort of a thing is actually advantageous for that bacterium. All right, some bacteria can actually produce certain kind of proteins and other molecules that can affect your gene expression. And that can allow them to establish their infection as well. All right, moving on to causing disease. Was that not part of it? No, that's establishing. Okay. Causing disease. And by disease, remember, deviation in health. So now a disease is going to be something where you actually notice that you are having an infection, or you have a fever or something like that. If you know something's wrong. Okay. Virulence factors associated with this. All right. Anything that allows it to cause an infection, the pathogen proper, and anything that allows it to establish itself in the host. So we have talked about some of these. Enzymes that allow them to get in between cells, get deeper into the host. That we talked about collagenase, right? Overreaction of the body defenses. We're going to go into that in a moment. And then altering host cell genome expression. Now, that's what we just talked about with the epigenetics before, making something that can control your gene expression. So we're going to get into the first two. First, you have to understand, again, about those exoenzymes. We learned what that meant in the last chapter. So enzymes that are secreted, and that includes any of these bottom ones down here, and things like collagenase, right? So mucinase, breaking down mucus that would normally catch you. Keratinase, that is, keratin creates a coating, a waterproof coating on your skin, not just your hair, but your skin, that keeps things from getting inside. So we can break through that. And then hyaluronidase, ACE enzyme, that's hyaluronic acid is not just part of our skincare regimen. It's also part of your extracellular matrix, which is why your body likes it in your skincare regimen. Okay. But yeah, so those are secreted and they help the microbe, and it's going to cause damage that you would see as signs and symptoms. All right. Toxins are anything that microbes or whatever would make that is poisonous to another microorganism. So it makes a toxin, a compound of any kind, and leads to damage. That's a toxin. Okay. One organism makes it and affects the other. Exotoxins, these are going to be secreted toxins. Usually these are going to be enzymes, but there's a whole lot of different ones. Okay. But the enzymes, exoenzymes, they often fall into that category as well. They're still toxins. They're causing damage. It's poisonous to you. Endotoxin. Now, I know we keep saying about endo being inside. So that would be toxins that are inside the cell, right? Yeah, sort of. Yes. But specifically the one that you should be aware of that we talked about in chapter 4, endotoxin specifically, that's a thing that gram negatives have. All gram negatives have endotoxin. That's the lipopolysaccharide, or LPS, on the outer membrane of gram negatives. I used to bring it up all the time, and now it's back again. Okay. All gram negatives have endotoxin. No other ones do. Here are some comparisons between exotoxins and endotoxin. See, it's still just one endotoxin. It's just one thing. But I'm not going to go over it. If you're interested, you can look at it. I don't need you to know the differences specifically. Just want you to know exotoxin secreted by organisms, endotoxin part of a gram negative outer membrane that can trigger the body to react to it with the immune system. Like it induces fever, for example. So this is an enzyme that breaks down red blood cells. We've seen this picture before when we learned about alpha and gamma and beta hemolysis. Enzyme that allows for that to happen, for hemolysis to happen, is an example of what we're talking about here, right? That would be damaging to you and your body. All right, inducing the excessive host response. Now, this is that next part of it. Some diseases are going to be caused by the body reacting inappropriately to the thing, right? So that tells us pathogenicity. how bad the disease and will end up being is not only determined by the microorganism you're being infected with, but by the host's response to it as well. And in fact, most of your symptoms and signs are going to be associated with your immune system responding to the thing and not the thing causing those symptoms or signs, okay? And it says down here, scientists speculate that the symptoms of long COVID, which can persist for months, can be caused by an overstimulated immune response to COVID. Right? So people who were overstimulated, that symptoms associated with COVID are going to continue to last for months because of how their body reacted to it, overreacted. Okay? Have you ever heard of toxic shock syndrome? Man, talk about an overreaction. You can die within hours. Okay. I already did click those. All right, local infections. Here we're talking about infections that will be in one area, like a boil or something. Systemic, all over the body. Focal infections started in one place and moved to one other place. It's focused into those areas. Mixed infections, you have more than one microbe involved. And then we have primary versus secondary infections. This is that example we could give. You got your urinary tract infection. It was your E. coli from your own bottom and whatever. And you cleared that infection usually through antibiotics, right? But now those antibiotics kill off your good guys in your urogenital region. And now you get a yeast infection. So that's a secondary infection caused by having had the first one, okay? We already did this. Let's just, okay. Acute infections. These ones are fast. They come on fast and they end fast. Chronic infections, they come on slow and they end slow. So that's pretty much, most of us are familiar with this. Signs, symptoms, and syndromes. These first two are directly related to one another, and we're going to go over what they are. A sign is an objective evidence, whereas a symptom is a subjective evidence. And what do I mean? Very important that you know the difference. Is definitely on the test, okay? A sign is anything that you can actually measure and observe and verify, okay? A symptom, it's subjective, right? The patient says they're having pain. How do you verify that they have pain at all, let alone how bad it is? That makes sense, yeah? So some examples, a sign that you can measure, fever, you can measure that. Swollen lymph nodes, you can actually palpate swollen lymph nodes. Vomiting, you know when that's happening, right? You can't hide that. You know if it's happening or not. That's a real thing you can see. Symptoms, okay? Pain, of course, we just mentioned. Fatigue, how tired are you really? I don't know, that sort of stuff. Nausea, so we have vomiting on one end, and nausea, you can't actually prove, right? So that's how we would separate these. The things you can actually identify and interact with versus the things that you have to just go based on what the patient is telling you. And if you go work in healthcare in general, you will be taught that you need to always believe what the patient is telling you. Even if they're just like coming in, yeah, I'm totally in the most pain I've been in my life. So can I get that, can I get that painkiller? No. Can I get the fentanyl, please? So, but you will be trained that you have to treat it as though it's a real thing. If you have repeat guys in there, you're usually going to be told to refer them to a clinic afterwards, but you have to treat them like it's real. Because how do you know? How do you make that decision? How do you draw that line? Of course, maybe you'll have a doctor that you'll work under that isn't quite as, forgiving. But so signs, you can measure it and see it, confirm it. Symptom, you have to just go on what the patient says. Now, syndrome, we stuck this at the bottom for some reason. It's not related to those two, except it does have to deal with symptoms. All right. If I tell you that you have a patient that came in with symptoms of sore neck, all right, photophobia, which is they don't like the light, okay, maybe fever, they're fatigued, malaise, that sort of stuff, and stiff neck, okay. Those symptoms all sound like meningitis, right? Right. Do you know how many things can cause a meningitis in general? How do you know? Just from those symptoms, that grouping of symptoms, can you tell that it is bacterial or fungal or viral, right? We don't usually think about that way, but they're all meningitis. They all have the same symptoms, but you have to break it down with your tests to determine exactly what's causing it. Like that Streptococcus pneumoniae, right? That's the more common one. If there's bacteria involved, it's likely it's that one. However, if it's bacteria that's involved and the person has petechiae, a rash all over their back, right? That person probably has Neisseria meningitis or meningococcal meningitis. Extremely serious, very deadly. It happens very fast. This is something where somebody comes in with those symptoms with that extra rash on top of that, they're going to pump you full of antibiotics right away and not wait for you to come back with confirmation of what you have. They're just going to shove it all at you because you could die that fast. You die before they diagnose you. They don't want to deal, so they give you all the antibiotics, and that's the right thing to do. And when they get the tests back, then they'll just narrow it down to the one you should be on. That's how serious that meningococcal meningitis is. Very serious. But all these meningitis, right? That's a syndrome. They all have the same symptoms. Another good one would be the common cold, right? There's 200 viruses that can cause the common cold, but they all have pretty much the same symptoms. Okay? Here are some examples of signs and symptoms. Signs that you can measure, right, and verify. Fever, septicemia, so how many organisms are growing in the blood, flourishing in the blood. Microbes in tissue fluids where they shouldn't be. Chest sounds, skin eruptions, leukocytosis, so a lot of white blood cells in something. Leukopenia, low white blood cells. Swollen lymph nodes, abscesses, tachycardia, the fast heart rate, and antibodies that are present in serum against whatever disease. Okay? You can verify all of those things. Whereas symptoms, chills, pain, malaise, fatigue, chest tightness, itching, headache, nausea, abdominal cramps, anorexia, and sore throat. Now, anorexia just means they don't want to eat. Okay. Those you can't really verify. You have to go based on what you're being told. So again, leukocytosis, increase in white blood cells. Leukopenia, decrease in white blood cells. Septicemia is going to be flourishing microbes in the blood where they should not be. Bacteremia, small numbers of bacteria in the blood. If bacteria is just there but doesn't seem to be flourishing, bacteremia. Okay. If it is flourishing, septicemia. So those two kind of extremes of one another. All right. Viremia, fungemia, those speak for themselves. All right. Some infections can be subclinical or asymptomatic or inapparent, right? You have the infection, but you're not having any symptoms. It's not manifesting disease. So no signs or symptoms. So you're not going to go seek medical attention and whatever. So most infections are going to be attended by some sort of sign or symptom. But there are a third of people who have COVID don't have symptoms. So they're walking around spreading around COVID without knowing it, right? That's important to be aware of these things. All right, now you've created your infection. You fought off all the defenses of your host and everything. You multiplied, and your little offspring that you've made are ready to go infect somebody else. Now they need to get out. This is the portal of exit. Okay. Obvious ones: coughing, sneezing, skin cells from open lesions as well, urine and feces, so waste products. We have removal of blood either by things like taking a blood sample or IV drugs or insect bites. Did you guys know? It's going to come later on in some of our other chapters, but it's going to say in one of the other chapters late on. But you can bleach your needles when you're doing your heroin, so you don't have to keep using dirty needles. I don't know what to say about it. I learned that last semester, and first time I saw it, I couldn't stop laughing. But anyways, yeah, bleach your needles, guys. Okay, be safe. So you can get out through all of the areas you might even think about, like mucus, sputum, nasal drainage for respiratory, skin, scales, cuts, fecal exits, urogenital exits, bleeding, ticks, fleas, other insects that will transmit your blood. All right. An infection that lasts for a long time, but might be hiding out. So like tuberculosis, right? You get infected and then it just goes quiet. And no symptoms, and you might not even know that you'd have had it. Latency. We learned about viral latency. Any disease can technically have some form of latency, whether it's just hiding out in your genes or hiding out in your cells or wherever else it's going to hide out. It's hiding out and it's latent. Okay. So most commonly thinking of tuberculosis, or things like chicken pox, just lying in wait to come back out. All right, next thing, sequelae. Or sequelae. I don't care what you call it, but to me it's sequelae because that's what it looks like. Okay. As long as you can recognize the word, man. long-term or permanent damage caused by infection that will last even after clearing of the infection. Did I talk to you guys about polio? I talked to you guys about that, right? About the iron lungs. Yeah. So that's a great example of something where you have something going on actively, but then some people walking away from that have permanent paralysis and stuff like that, like FDR. FDR didn't have polio while he was president. He already had it, but he was paralyzed from it. So he would hide, like put his blanket on it to hide so that nobody would know or think about him having polio. But he was not actively sick, right? So that's sequelae, something that lasted after the infection was gone. Like it says here, strep can lead to rheumatic heart disease. I don't know if you guys know about rheumatic fever, but it's basically the strep looks like has molecules on its cell surface that look like your own cells. And if your body reacts to it and also happens to have antibodies that then will also react to your own cells, it's like autoimmune, basically. And then that leads to rheumatic fever and rheumatic heart disease as a result of the damage associated with it. Meningitis can lead to deafness. Lyme disease can cause arthritis, and apparently like everything you could ever have wrong with you can be caused by Lyme disease is what I hear. And then polio, of course, paralysis, or even other issues, neuromuscular issues. All right, courses of infection, how it progresses, right? We have the incubation period. This is just the period of time that we've been exposed to the microbe, and it takes for it to grow enough that you start having symptoms, okay? That period of time. So it has to grow in order to start doing its thing. That's incubation. To get exposed to the flu, and 2 days later, you typically will start showing symptoms after you've been infected, because it takes that long for enough virus to be affecting you as a disease. Okay, so that's the incubation period. They're basically growing period, essentially, for lack of a better explanation. Prodrome, the prodrome or prodromal period. Let's just go to this chart. So incubation period, we're just growing, no disease yet. Just got introduced, just growing, no disease. The prodrome or prodromal stage, this is the phase where you have your first symptoms, like a fever. We wake up one day with a fever. You're like, oh, good, this is great. That's the prodrome, all right? You know what's coming after that. Maybe it's going to be a whole lot of coughing and pain and, you know, being tired and all that after it. While all that mess is going on, the coughing and the pain and whatever, this is at the height of your infection. So we call this the acute phase. This is the worst of it, okay? And then after you start getting over your symptoms and starting to feel better, this is convalescence, or the convalescent period. Convalescence. So those four is the ones that I need you to know. Don't care about that, okay? Those four: incubation, then the prodrome, your first symptoms, the acute phase, the worst of it, convalescence, you get better. All right. Where pathogens exist whenever you're not sick with them, or other people you know aren't sick with them. The reservoir. This could be other animals, or it could be other populations, maybe. Maybe this population has cholera all the time, and now that it's been moved over to America temporarily, then the reservoir would have been, you know, the other population that has it. Maybe, you know, Haiti or something like that. Like that was the issue for a little while. Or animal, soil, plants, anywhere like that. Those are the reservoirs where it normally exists. The source is going to be the individual where the infection was acquired to the rest of the population or group, right? If you did happen to travel to Haiti and you got cholera, and now you're like sharing your water with everybody, and they all get cholera, then you would be the source. Okay. All right, carrier is somebody who is inconspicuously sheltering the pathogen, usually without knowing it, and spreading it, usually without knowing it. There's different kinds. Asymptomatic carriers, they don't have any symptoms, they're just infected. This is what I was talking about with COVID, right? People not knowing. Or we could even be talking about typhoid Mary. Does anybody here know typhoid Mary? What that is? Like sometimes people, like you'll be walking around coughing and whatever. Oh God, I got to feel so old when I say these things. But they'll be saying things like, man, you walk around like typhoid Mary. And what it means is you're spreading your disease around to other people without like caring about it, okay? Typhoid Mary, she was fine. She was an asymptomatic carrier of a disease, right? She liked to cook, and she would cook for soldiers in the hospital she worked at. And she loved doing that, interacting with everybody, except people started getting sick and dying, okay? They're getting sick and dying. And they found out through some tests and stuff and symptoms of the people involved that she had to have a form of salmonella called typhoid fever. Okay, it's like dysentery. Okay, it's like one of the worst kinds of salmonella you can have. She was just carrying it in like her bile duct or something is where it was living, but it was enough bacteria that she was spreading it to people because she didn't understand about washing her hands and all this. So she was spreading it. People were dying. They told her, hey, man, you need to wash your hands, number one. Number two, you need to stop cooking for people. You can't do this as like your job anymore. You have freaking typhoid fever. Okay. And she was like, okay, yeah, okay, I can do that. I can do that. So she moved to some other place and changed her name and kept doing it. So more people were dying. And when she was caught, they imprisoned her. I won't say imprisoned, but they confined her for the rest of her life, which ended up only being 15 years after that. But that's Typhoid Mary. They consider her a public health issue because she was carelessly, passing this disease around that was killing people. So, right, that's Typhoid Mary. Now you know the story. All right, then we have people who are incubating carriers. You just got infected with it, but you don't have symptoms, but you have enough virus to spread around. Okay. Convalescent carriers, the opposite kind of other end of that. These people who are getting over their illness and are still contagious, right? So this would be people who often I have found like to have a cold or a flu or COVID, and they feel better now, but they're still coughing a little bit. I feel good enough to go back to work. I feel good enough to go back to school, and they come back and they're not completely over it, and they're giving everybody flu or COVID or whatever. That's convalescent carriers. Chronic carriers, people who are carrying around like things like tuberculosis or like Typhoid Mary. I guess that's a better example. But they have this thing they don't have any symptoms of, and they're carrying it for a very long time. And then passive carriers, often this is going to be medical and dental personnel who will be in contact with things and could be potentially, they're not being careful, transmitting from one patient to the next, that sort of stuff. Okay. Living reservoirs, animals, or actively ill humans can transmit diseases. That shouldn't be a surprise. We're going to come in a moment to indirect and direct transmission. I'll come back to it. And then human carriers just in general, things like carriers of HIV who may not even know that they have it yet. Arthropods, these are things that drink our blood and transmit things that way, like malaria, right? A vector, this can be biological or mechanical in nature. A vector is something that is going to transmit an agent from one host to another. Biological vectors, they have to be infected with that thing, okay? The mosquito being infected with malaria. Chickens, but I would think of, it could be salmonella, right? Or it could be the flu. And then here's this bat. Well, we know so many diseases actually do come from bats. We believe that's where Ebola came from and COVID, the original version. Anyways, that's what they say. Anyways, mechanical vectors are not infected. So the cockroaches and the flies, we know how they work. They're going to be walking across the nasty and then come and land on your food and transmit the nasty to you that way. So they're mechanically giving you the disease, but they're not infected themselves. A zoonosis is anything that exists in animals normally that's getting spread to people, like where it shouldn't normally be, like rabies. That's, I think, the best example. Everybody knows what that is and where it comes from. Animals. So rabies. Yeah. Toxoplasmosis and all that. So there's some lists of zoonotic infections if you're interested. Non-living reservoirs, places where you can get these organisms, like from normally they exist in this water, normally they exist in the soil or whatever it is, okay? Outside of person-to-person transmission. All right, communicable disease. This is any disease where you can transmit the infection from one human host to another human host, okay? It's communicable. It can be transferred from one to the other. However, if it's highly communicable, very highly, especially, like it says, especially through direct contact, which I'll get to in a moment, then that's contagious. So obviously, COVID is contagious. The flu is contagious, right? Things that may be communicable wouldn't be quite so easily transmitted, even though contagious still falls under communicable. Yeah, it's just really communicable. Non-communicable diseases, these are not going to be allowed for transmission from host to host. In the case of people to people, if you have malaria, I can kiss you all day long and not get your malaria, right? So that is a non-communicable disease between humans. Horizontal transmission means transmitting something from person to person, essentially, unless vertical transmission is mentioned, parent to offspring. So that could be ovum, sperm, placenta, or milk. So some sort of parental relation in vertical. Okay. And horizontal, everybody else. Okay. So spreading from within the population via direct contact, which could be me sneezing on your faces right here. That would be direct contact, or indirect transmission. So if I'm up here and I leave my soda up here, and one of you is like, oh, I love a Coke Zero. Oh, it's just so hot in this place. I need a Coke Zero. And I left it up here, but I have COVID, and you drink it, and now you get COVID, right? Normally, most normal Coke Zero cans aren't going to have COVID on them, right? Not naturally existing Coke Zero cans, but right, they're free of COVID typically. I had to introduce it to it in order for it to get transmitted to you. Then this is called a fomite, which is a particular kind of vector. Okay, so it's transmitting the thing for me. It's indirect, and this particular instance where this has no infection associated with it at all, otherwise, is a fomite. Okay, these are terms we need to be aware of. Yeah, it could be, yes. In that particular case, yes. All right, so we already mentioned vehicles. We're going to transmit with something else, not direct transmission. Fomite is not a continuous source of infection. That's the kind of the specifier here. It wasn't, and it will not be after it gets cleaned, right? Oral fecal route, we kind of know about this. You need to wash your hands when you go to the bathroom. It's really where most of this comes from. Or drinking water that's contaminated, I suppose. All right, so we know water, soil, and air can be vehicles as well, especially if we're talking about sneezing and coughing. You have droplet nuclei and aerosols. So microscopic pellets of mucus and saliva versus suspensions of fine dust particles. They can like chill in the air for hours. And I might cough up here, and then I don't know, Dr. Tim comes in here a couple hours later, and he gets my tuberculosis because he walked through my tuberculosis air. It's crazy to think about. All right, next we get to Robert Koch. Robert Koch, he developed these postulates called Koch's postulates, go figure. He used these to determine the causative agent of anthrax. And here's how they work. He would take a sample from a patient who he believed had this disease and isolate the organism that he believed was causing the disease. Now he's out that pure culture, he'll introduce it to another specimen, in this case, a mouse. It has similar symptoms and dies also. And so then he is able to reculture out of that mouse the same thing that he started with. So he's shown that that is what causes this. That's how he proved about anthrax. That's how he did that. Koch's postulates. An etiological agent, by the way, is just the causative agent. That's what that term means. I don't know what to say about this. Yeah, of course, we have a much more updated version of this now with molecular biology. No surprise there. All right. This isn't written correctly, but I'll tell you what it should have said. It should have said, give an example of a disease and list associated signs and symptoms. Definitely start thinking about this sort of thing as the test is a week away.